Is it possible to put it on YouTube?
Good evening, ma'am.
Good evening, sir.
Good evening, ma'am. Good evening. Nice.
Sir, you can start now, sir.
Yes, ma'am. Yes, ma'am. Thank you. Thank you. Hi and welcome to wonderful session of the biological classes. Good evening, my dear students. Today, what we are going to discuss about, the part of the biotechnology, particularly the obtaining the foreign gene product. How will you obtained the foreign gene product in laboratory aspects? That things we are going to discuss deeply. And then how will you produce it? Okay, in large scale. Suppose taken examples, any one of the microorganism we need in large amount in laboratory aspects, that things also we are going to discuss with the help of the bioreactors. What is meant by bioreactors? How it will be used? How many parts is there? Okay, that things we are all going to discuss in future slides. Okay, so now, we are going to discuss the obtaining the foreign gene of the product. Take an example. When you insert a piece of alien DNA means another species or any one of the species into a cloning vector and transfer it into a bacterial plant or animal cell, that alien DNA gets multiplied. So that things already we discussed. So when you are inserting, when you want to insert the new gene, when you want to produce any one of the new genes, is it cut the DNA? They all are already detailed, isn't it? Then only you will get the, what is that? New DNA, new genes we can obtain from the laboratory. See that one in almost all recombinant technologies, the ultimate aim is to produce a desirable proteins. That is very, very important. So the end of the gene product or biotechnology or genetic engineering, whatever it may be, what will be the ultimate aim? Means you should produce a desirable proteins only. For example, take an example, immunoglobulin. So one of the protein molecule, isn't it? We cannot be able to synthesize artificially. Okay. That time what we have to do, we have to select the classmates, those who are all responsible for the production of antibiotics or immunoglobulins. Okay. That gene first you should identify, then you have to isolate, then the process already available. Okay, the hybridization process, PCR technique, and also insertion, elongation, and also that process also you have to do. After that, you will obtain the combined form of the DNA molecule, isn't it? So that combined form is also, what is the main use of that one? The particular protein biosynthesis is very, very important. That after the biosynthesis, what happened, it was the ultimate aim. It will be the ultimate aim. Hence, there is a need for the recombinant DNA to be expressed. That is very, very important. Then only the desired quality, this desired quality of gene produce, particular DNA molecules produce the protein molecules. Already we know about that things also. Okay, by the process called as translation. Okay, then what happened, that protein molecule used for the commercial purpose. No, they may be used for the society. That is the very, very important one, my dear. What the next one is, the foreign gene gets expressed under the appropriate conditions. The expression of foreign genes is host cell involved understanding may technical details. One more is that when you are selecting the foreign gene, that may be gets expressed under the appropriate which condition it will be acted. That things also we gather the knowledge from the particular method, and also trial and error also. Okay, what are the condition, what are the temperature, how it will be, which one is the primer. So all those things we should know. Okay, by this technique only. And one more is that the expression of the foreign gene in host cell involve understand many technical details also. Okay, after having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing in a large scale. Okay, so that is very, very important. That is very, very important idea. Okay, see that one. What the next one is, the protein encoding gene expressed in heterozygous host, heterologous host. For recombinant protein, the cell harboring cloned genes of interest may be grown on a small scale in the laboratory. Small scale means it may be the particular amount will be microgram, microliter, millimicrogram. So likewise, they may be obtained. Suppose if you want to produce more amount of protein molecule in laboratory in large scale, it has a specific technique. Okay, we will see the next slide. The culture may be used for the extracting the desired protein and then purifying it by using different separation techniques. Okay, already you know about when you are preparing any one of the protein molecules. After that protein molecule, what you have to do, you have to purify. After the purification, okay, it may be commercialized. Okay, during the purification technique, there are so many biomolecule technique, biotechnique is there, biochemical technique is also there. Particularly taken examples amino acids, a paper chromatography is there. Even taken examples of protein molecules also there. Suppose if a mixture of protein, if you want to isolate, if you have a mixture of protein molecule, if you want to isolate, so that time what they were using, the one of the technique is called as, what is that, chromatographic. There are so many chromatographic techniques also there available in commercial aspect. Paper chromatography, thin layer chromatography, and also beads. Okay, so these are the one dimensional, two dimensional. Okay, they are all are essential for the purification technique. And then electrophoresis. Electrophoresis is most important. Okay, they were used the gel electrophoresis, a polyacrylamide gel electrophoresis. The gel electrophoresis not in that, and also liquid chromatography is there, and solid chromatography is there, and then thin layer chromatography is the TLC, and then another one is called as HPLC, high pressure liquid chromatography. These are all the bio technique. They were used for the purification of the biomolecules. What are the biomolecules? Already we studied, protein molecule is there, lipids is also there, carbohydrates, enzymes. Okay, and particularly protein, isolation of the protein. Take an example, there are so many fractionation is there. Suppose if you want to separate the molecules, I will show you, it's better for your understanding. Okay, so there are so many biotechniques that were used for the separation of molecules. Paper chromatography, one is called as a technique. What are the technique? So there are so many techniques that were used. Okay, so first of all, paper chromatography, paper purification process, a paper. Okay, paper chromatography, paper chromatography. Okay, so paper chromatography, simply called as, another one is called as TLC. So this is denoted as, what is that, thin layer chromatography. What does that mean? Thin layer, THIN, thin layer chromatography, thin layer. Okay, layer chromatography. Okay, so same things is also there. Thin layer chromatography. And third one is called as HPLC. Already told you, HPL and C, HPLC, high pressure liquid chromatography. Here they were used as a liquid. Okay, that is called as one of the very important procedure, the mostly used for the biochemical aspect in the hospital as well as any other bio laboratory. Okay, high pressure, okay, so high pressure okay, high pressure liquid. Okay, liquid chromatography. Okay, liquid chromatography. Okay, simply C denoted as chromatography. and solid chromatography is there. Solid chromatography, liquid chromatography, gas chromatography is there. Okay, so these are the different type of technique that were used for the, what is that? Purification of the molecule. What is that? Purification. 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 Okay. Purification. Okay. So this is very, very important. Purification of, what is that? Biomolecules. Okay. By the technique is called as biotechnique. Biomolecules. Biomolecules. What are the biomolecules already studied? You know about the major one is the protein. Very, very important, because already you know about protein. Okay, the protein is very, very important. And second one is called as lipid. Okay, lipid, otherwise called as, simply called as, what is that? Fat. And then third one is called as nucleotide, nucleic acid, DNA and RNA. Okay, that things also. So nucleic acid, simply we may call it as a DNA. Okay, and then RNA. Okay, so RNA, DNA and RNA. And then, so take an examples of enzymes. Enzymes. Okay, enzymes also already in our, but very, very important. Enzymes, suppose if it is pure form only, it act upon the substrate. Enzymes. Addition with this, there is a hormone. There is a hormones. So these are the biomolecules. We can, what you have to do? Hormones. Okay, these are the things. Okay, these are the biomolecules. We are going to separate by the technique is called as biochemical technique. It's a separate, what is that? Subject in your book when you are going to study it. Suppose if you want pure forms, how will you, just take an examples. The plant cell, it contains the mixture of component. That mixture of component that can be easily, that is not easily to separate the molecules. So that time they were used the special technique. The technique is different according to their molecules. What are the biomolecules? For example, protein and enzyme, both are same. Okay, because it's a enzyme is made up of protein molecule. Okay, the protein is also is made up of which one, already about one of the important component. Which one is that? Amino acid. Amino acids. So more than one amino acid together form as a protein, isn't it? The protein all are made up of which one, enzymes. Lipids already but lipids, there are so many lipids there. Okay, simply we may call it as fatty acids. Fatty acid, different types of fatty acid is there, and essential fatty acid, non-essential fatty acid. Likewise, the enzyme is also essential, amino acid, essential amino acid and non-essential amino acid. These are the things also there. DNA and RNA, not only the DNA and RNA, and plasmids also there. Hormones, here you may have to split into two different main category. Okay, main category. Hormone is a plant and element, plant and animals, plant hormones as well as the animal hormone is also there. Animals. So the plant hormones, okay, what are the plant hormones? They are all are essential for the growth as well as the, what is the incubation activities. Likewise, the animals also. Animals already in our world, but higher animals and lower animals, particularly human beings. Human beings, and there is a two things is there. One is called as male, another one is called as a female. Male hormone, female hormone, and also common hormone is also there. Already you studied these things. These are all if you want to isolate, isolate and also identify, isolate, and purification also done by the this technique only. After the purification, they may be used as a which one, commercial purpose. Take an examples of immunoglobulin. Immunoglobulin comes under the getting known as which one, protein molecule, immunoglobulin. Okay, it is also comes under the getting of antibodies, AMB, isn't it? Antibodies. Not only that, and all the protein molecules already you studied. So many protein molecules, a simple protein, complex protein, derived protein. Likewise, it comes, isn't it? So simple protein, albumin, compound protein already in our report. Okay, keratin. Okay, these are so many protein molecules also. And also derived protein is also protein and protein derivatives, all those things. Lipid is also fatty acid and fat, glycerol and fatty acids also. DNA already in our report. Okay, it depends upon the micro nucleotide. Okay, nitrogen base, they can easily isolated from the this technique. Okay, my dear sir. Okay. So these are the very, very important technique, particularly in Biotechnology, which technique they were used? No, so many techniques they were used, particularly PCR is also one of the application process, and also that hybridizations. Okay, genetic engineering is also one of the best. Final result, what is that? Protein. The particular DNA molecule responsible for the production of particular protein molecule. That particular protein molecule, after that isolation, what you have to do, it's commercially prepared in large scale. That is very, very important. Final ultimate end. That can be introduced in the market and commercial money making process. That is a very, very ultimate. Okay. So that's all things. Okay. Go to the next one. See that. See that. So large scale in lab. The culture may be used for the extraction, the desired protein, and then purification. It by using different separation techniques. That is the different separation techniques already we discussed. Okay. So the protein encoding genes expressed in the heterozygous cause. Okay, already we discussed these things also. See here. The cell can also be multiplied in a continuous culture system. This is also another important continuous culture system. Okay. System wherein the used medium is drained out. That is very, very important. Okay, drained out. So we cannot use the same culture medium. You would not use the same culture medium, because once you use it, what happened after the separation, it can be discarded. Okay, from one side, while fresh medium is added from the other, maintaining the cell in the physiological most active lang and exponential phase. This is very, very important, my dear student. We cannot be able to use in the take an example. Any one of the land also we are using each and every time we are what is that doing some process. Okay, see that one. I will show you how it will be. This one. Okay. Next one is suppose the medium. Okay. What is a medium? Agar medium already in our, but there are so many techniques that were used. Maximum petri dishes already we discussed. Petri dishes nothing but one of the small disc-like structures. Disc means a small plate only. Okay, this is called as petri plate, and well about the test tubes. Okay, test tubes, different size and shapes is also there already. Well about test tubes. Okay, so this is also another one of the things. And third one is a conical flask. Conical flask. Okay, so for example, this is the conical flask. The size is also vary. Already about, isn't it? The size is also vary. Okay, so this is another type also there. Okay, now so here what happened, there is a medium is that medium essential for the growth of the any one of the microorganisms. Take an example, this is the pot. Okay, so this is a growth medium. Growth medium already we discussed, depends upon the sources what we are going to isolate. Okay, so this is the petri plate method. Okay, these are the things. This is the first one, and second one, and third one is also that. Okay, here also that. Here also there is a sometimes they maybe grow like this. Okay, here the nutritive medium is there. Here the nutritive medium, that nutritive medium containing all the nutrition for their growth of the microorganism or plant or whatever it may be. Okay, so what is this? These are all called as, what is that? Nutritive medium. Okay. Okay, nutritive medium. Okay, so whatever it may, you can imagine. Okay, once you culture any one of the microorganism in the first culture already there are so many serial dilution is also, serial culture is also there. Suppose take an example in this plate, in this technique is also, imagine this is the first one, you can take another petri dish for the second one. So another petri dish for the third one. So this is the second one, third one, fourth one. Okay, likewise you can use it, isn't it? But we cannot use the same nutritive medium for the further growth also. We cannot be able to use the see that. So this is considered first one. Second one means just like this. Okay, second one means you have to put like this. Okay, so this is the second petri dish, the second one. Okay. Okay, from this one, yeah, this plate, this petri plate, we should select the desirable microorganism from here to here. We may culture here. So this is called a subculture. Okay, this is called as what is a subculture. Don't forget. Okay, subculture. Okay, what about this one? This one is called as mother culture. Okay, this one is called as mother culture. So these are all are called as mother culture. Okay. So from this one, subculture to here, we are going to the next one, third step. So subculture. Okay, subculture. Okay, this is a subculture. Subculture. You know the third one. Okay, just imagine. So the purification of the first one compared to the last one. Which one is the highest one? You know, the purification comparison between these three. So this one is the medium one, this one is the 50%, this one is 100%. Likewise, we have to maintain all these three. Okay, here also you have to place it. So many small petri dishes. Okay. This is the culture from here to here, again here to here, and here to here. So this one is a good quality. Okay, this is another method. Here you can imagine same process is also there. Okay, so same process we have to do the another again culture. Okay. So this one is from here to here, and here to here also. So this is a mother culture. After that culture, after the complete the culture, what happened? You have to take the sample from this one, first one, and here we are going to introduce the next conical flax. This is called a subculture. Already we discussed, and third one is subculture. Okay. So here we cannot use the same medium. You have to replace the medium here. In this part, we are using the new medium only. Then only the growth will be in pure conditions. Suppose we are using the same medium, what happened now? It will not. You will not obtain the good result. That is very, very important. So you have to change the medium. Okay, that is very, very important, my dear students. Okay, see that. See that. That's it. That is the things given here. So the cell can also multiply in the continuous culture system. When the used medium is drained out from the one side, while fresh medium is added from the other maintain, the cells in the physiological most active longer at exponential phases. This type of culturing method procedures, okay, a large biomass leading to the higher yield of desired protein. That is very, very important. When you are using the new medium, okay, so that is the one of the procedure for the production of large biomass leading to the higher yield. Higher yield, if you want to know, you have to give more nutrition for their particular microorganism or what is that? Species. Okay, then only will get the good yield. Likewise, just like a plant. Okay, it's also one of the harvesting methodology. And small volume culture also cannot yield appropriate quantity of the product. That is very, very small volume culture. Small volume means microliter, millimicroliter, and so on. So that cannot able to produce less amount. That is very, very important. That is not a good quality. Suppose we are taking a 10 ml, it is culture means you will get the yield will be very less amount. Suppose 100 ml, you will get 10 ml. 1 liter, you will get 100 ml. Likewise, you can imagine. Suppose if it is the volume will be very less, the product quantity is also very less. To produce a large quantity, the development of the bioreactor, don't forget, my dear student, this is very, very important. If you want to grow a microorganism in the large tank, okay, we were used one of the huge instrument. That huge instrument is called as bioreactor. The volume large will be 100 to 1000 liters. For example, take an example, ethanol production is one of the commercial purposes, isn't it? The ethanol is also prepared in commercial, in particularly sugar industries. Okay, it will not produce 100 ml or 200 ml like that. They will produce only 10,000 gallons. Okay, 10,000, 100,000. Okay, likewise, millions gallons also produced. There are so many things. Take an example, CAD industries, ethanol industries, and also antibiotics also. Okay, and particular protein molecule. Okay, and so many protein, yogurt protein is also that commercial work, dairy protein, casein protein, all the protein molecules, they were used as different type of instruments. That is called as uh, tank. Okay, and already you studied in the ARS, that one sewage treatment tank also. But here, what is that? This is also already they are asked the question. NEET point of view, they were asked the question. This, okay, the large quantity of the microorganism produced by the one of the instrument is called as, what is that? Bioreactor. Don't forget, my dear student. This is very, very important. Okay. See that the structure of bioreactor will be like this. Okay. See that structure. There is a one of the small, what is that? It's completely covered a steel plate, completely covered. Within the steel plate, there are so many rotator is there. Each and every rotator, there is a specific blade also there, flat blade in power. And then inlet outlet is also there. Then maintaining temperature also indicated, and also the outlet is also here. Is it? This is also a motor. For the inner, there is a propeller. Propeller is that the propeller is rotate itself by the action of motor. This motor. Okay. So here acid base for the pH control. This places, there is a acid base. Suppose here acid is there, we are adding the base. Suppose it indicate the pH will be very less, we are adding base. Suppose the pH will be very high, you are adding the acids. Okay, then only it will be maintained. And the stream for the sterilization. Suppose the container should be sterilized before sterilization. Okay, they wash the complete process with the help of soap water or whatever it may be. After that, washing process finished, steam for sterilization. So completely eradicate the microorganism. It will not kill the microorganism, just like a pasteurization. Okay, after that, they were introduced the mixers. Okay, flat blade or an impeller. So these are the things. See the next one, you can easily identify. This is the large scale industries. They were products the bioreactor. See how many bioreactor is there? 1, 2, 3, 4, 5, and that side also having. Okay, so many things there. It's a big process, commercially prepared. Okay, and then next one is, see that this is the structure, the sparge tank bioreactor. Okay, some of them are some modification here. Okay, they increase the surface area for oxygen transfers. This part. Okay, this part. So here the rotator, already you know about. The gas entertainment here. The gas is also produced. That gas is also produced. The bubbles dramatically increases the oxygen transfer area. So these are the another process. This is the simple process. Okay, diagram is given for the bioreactors. That things also. See the comparison between simple stirred tank bioreactor and the sparged stirred tank bioreactor. These are the two reactors have been given. One is called a simple stirred tank. Okay, and another one is called a sparged stirred tank. So comparatively these two things, what are they? Here there is a small blade only is that both the side, but here each and every steps there is a blade is there. Flat blade is also there. That is that blade is also used for the mixing of the container. What we are giving introducing, then only the microorganism it will be grow, it produced. Okay, likewise. See that one. Thus, bioreactor can be through of as vessels in which raw material or biological converted into specific product. That is very, very important. So what is the main uses of the bioreactor? Means raw material. Take an examples. One of the very good examples. Okay, don't forget, my dear, this is very, very important. Okay, so many times they were asked the questions. Okay, see that one. Okay, take an example, CAD making process. Okay, CAD making. Okay, so another one is a, take an example, CAD making. How it will be possible, pasteurized milk, pasteurized. Okay, pasteurized. I will put the here. I will pasteurized. Pasteurized. Okay, pasteurized milk. Okay, pasteurized milk addition with this curd. Okay, so what will be the result? Already you know, but what will be the result? Curd means it's nothing but the microorganism. Addition of the microorganism. It's a small amount of microbes. Okay, microbes maximum already you know about this is the how will it be? The small amount of microbes. Okay, microbes means what is that? A small amount of curd we are adding. The curd containing what is it? Microorganism. Which are microorganisms mostly present in the lactobacillus. So as a result, the particular temperature, a complete large scale it will produce. Suppose take an example. Milk is a substrate, isn't it? Milk. Say here, milk is a substrate. Milk. Just to take an example, milk is a substrate, isn't it? So from the milk, we are adding a small amount of curd, otherwise called as microorganisms. Microorganism, that is called as culture. Microbes. Okay. Culture. The culture containing microbes, isn't it? That is lactobacillus. Which one is contained, lactobacillus. Lactobacillus. You can take an examples. Lactobacillus. The lactobacillus. Okay. The lactobacillus is a microorganism act upon the milk. Milk contain what is that in milk? Sugar is nothing but the lactose. Okay, lactose. So here the lactose, milk containing lactose, the lactose is the milk sugar, isn't it? The lactose is a substrate. The lactobacillus is nothing but the microorganism that produce the enzyme is called as lactase. That enzyme is called as lactase. Okay. Okay, lactase. Okay, lactase. The lactase, what happened, automatically act upon the lactose. As a result, it will produce the lactic acid. Lactic acid is the end product, lactic acid, by the action of lactobacillus. Here, lactic acid only. Okay, acid. Okay, this is commercial oriented. Lactic acid will be produced. Once again, repeat for you. Milk contain lactose. Already know about. It's a milk sugar, otherwise called as what is that? Disaccharide, isn't it? Lactose. The lactose always the microorganism lactobacillus act upon the lactase as a substrate. The lactose converted into lactic acid by the action of lactase. Okay. Once again, repeat for you. Okay. Once again, repeat for you. Already know about milk containing lactose is a sugar, isn't it? The lactose, how it will be converted into lactic acid. See that? Lactose, how it will be converted into lactic acid. Lactic acid. ACID, isn't it? Lactic acid. How it will be converted, by the action of lactobacillus. Lactobacillus. Okay. Lactobacillus. Okay. The lactobacillus produce one of the enzyme is called as lactase. Okay. So ASC, lactase is an enzyme. The lactose is the substrate. Here the substrate lactose. The lactase is the enzyme act upon it. So the lactose converted into lactic acid. So it is commercially oriented. Here how much amount they may be produced, it is not a 100 ml. Okay, it is not a 100 ml. It may be a 10,000 ml or 1 lakh gallons. Okay, gallons. That much amount they can produce in commercial oriented by the action Likewise, the ethanol is also, ethanol already know about, what is that? Sugar molecule. Take an example. So another is examples also. So another example is ethanol production in commercial and penicillin production and antibiotics. So many things is also there. Okay, they are all are commercial. Ethanol alcohol, methanol alcohol. Even take an examples. Liquors. Okay, take an examples. All are, it has a specific. Okay, see that one. Commercially prepared. They are all the large amount they will be produced. So next one is got as what is ethanol production. Okay. Next one is ethanol. Okay. Ethanol production. Ethanol production. What are the raw materials? One of the raw materials, sugar molasses. Otherwise, called as what is sucrose. Okay. Sucrose. Sucrose is nothing but it is also one of the disaccharides. Okay. Sucrose. Plus, what is that? Yeast. Okay. Yeast is one of the unicellular fungi. Okay. So as a result, what happened? You will get, what is that? Ethanol. Okay. Ethanol plus, what is that? CO2 you will obtain, carbon dioxide. Okay. Here, what is the action? What is the action? The yeast, what happened? They produce two types of enzyme. The two types of enzyme, what is it? One is called as, what is that? Invertase. Okay. Invertase. Okay. Invertase. Another one is called sucrase. Okay. So act upon it. Invertase, what happened? The sucrose molecules converted into, what is that? Glucose plus fructose. The conversion of this one. You act upon it, as a result, this one. After that, the glucose, what is that? Converted into ethanol. Okay. This is also one of the another methodology. They were used for the large scale industries. That is also very, very important, my dear students. Don't forget this one. It's also very, very important. See that. And bioreactor can be through the as a vessels in which a raw material already told you. For example, curd making industries, milk is a raw material. At the same time, the ethanol production means sugar molasses is also one of the raw material. Biological converted into specific products. Individuals enzymes, that is very, very important. Individual enzyme, lactic acid, how it will be produced? Lactose is a substrate. Lactase is an enzyme. That lactase enzyme secreted by the, which one lactobacillus. So the lactose only act upon the lactase enzyme only. The lactase enzyme only act upon the lactose milk raw material only. Ethanol is also, for example, so many common starches also, amylase, amylopectin, lipase, so many things is there. Particularly using microbial plant and animal or human cells. Plant means, some of the microorganisms, such as lactobacillus, streptococcus, penicillium, yeast, all those things, they are all are considered microbial. Okay. And plant and animal sources also, they obtain the enzymes. That enzymes all act upon the particular raw material. It will produce a, for example, Roquefort cheese. Already about. Okay. Roquefort cheese is also the carbon dioxide, the cheese size will be, the carbon dioxide relieved, the coal will be very large in nature. What is that reason? Due to the presence of the microorganism, it emits the carbon dioxide through this one. Take an example of bun. Okay. It is one of the fermentation processes. How it becomes, inside there is a small sponge-like structures. After the fermentation date, what happened, when you are placed into the oven, what happened, how it will be in the spongy nature. After the fermentation, what happened, it contains carbon dioxide, isn't it? The carbon dioxide, when you are heating, carbon dioxide will be weightless. So it will be eliminated from the floor. Okay. So as a result, it make a small hole like such. That's why the hole inside is there. Okay. That is very, very important, my dear student. And the next one is the bioreactor provide the optimal conditions for the achieving the desired product by the providing optimum growth conditions, temperature, pH, substrate, salt, vitamins, and oxygen. This is very, very important. Each and every growth, it has a specific temperature. Okay, some of them are they will be grow low acid condition. Some of them are they will grow in the high pH condition, also high temperature. Okay, you know about that optimum temperature. CAD making process also done by pasteurization is nothing but 60 to 70 degrees Celsius, 65 degrees Celsius. Suppose eradication, if you want to utensil or any other equipments, if you want to sterilize it, you have to give up to 120 degrees Celsius up to 30 minutes. Then only the utensil or any other equipments will be completely sterilized. Okay, that is also very, very important. pH is also very, very important. Some of the growth culture, they can grow up to the 6.5 to below 7. That is acid pH. Some of them are neutral. Some of them are more than 7. That is what is base pH. Okay, that is also very, very important. Substrate salt. Substrate salt means what is that? When the enzyme activate upon the any one of the medium, that medium is called a substrate. Take an example. Lactose is a substrate. Lactose is a substrate salt. Okay, they act upon the enzyme lactase. So the enzyme substrate complex will be formed. As a result, the product will be lactic acetic acid. Okay, already you know about it. Acetic acid is also one of the very, very important things. Okay, and butyric acid is also production by the commercial aspect in large scales. Okay, they were used the specific microorganisms. So that microorganisms produce particular enzyme. That enzyme only act upon the particular substrate. Okay, and then vitamins and also oxygen. Vitamins is very, very essential. Some of them are they will grow very rapid nature. Okay, the culture. Suppose there is a acid production tank is there, no? Suppose you are adding some small amount of base, what will happen? No? Immediately the acidity will be very high. Okay, opposite reaction will be get it. Okay, and also oxygen. Some of the microorganisms are anaerobic, some of them are aerobic. Aerobic means you know well, but they need the microorganisms. They will grow with the help of oxygen. Such type of microorganisms are called as aerobic. Anaerobic, absence of oxygen. They do not need which one you know. No need of oxygen for their growth. Okay, some of them are facultative. Facultative means whether presence of oxygen, whether absence of oxygen, they will grow. Okay, these are all very, very important. And also the most commonly used bioreactor of stirring type. Stirring type, what does that mean now? You have to mix properly all the substrate used by the microorganism. Okay, suppose some of the tank, okay, we are introducing the substrate, they may be settled down. Okay, so you will not get the proper result. So it will be continuously mixed with the stir. Okay, so this is the common. All the bioreactor are particular temperature, pH, substrate, salt, vitamin, and oxygen is also not only that, and also there is a stirring type only, continuously stir. Then only the uniformly mixing of the substrate with the enzymes. So as a result, what happens? You will get the 100%. Suppose if it is not properly mixed, you will not get the proper result. That is the reason. That's why they were used the bioreactor of stirring types. Okay, so go to the next one. The stir tank reactor is usually cylindrical or with the curved base to facilitate the mixing of the reactor content. That is the reason. That's why I told you. The stir facilities even mixing and the oxygen availability throughout the bioreactor. Some of the microorganisms they need oxygen means what happened, you know? It has a separate pipe for the pipe to connect the tank. Okay, the sublimination of oxygen is also very, very important. Some of the bioreactor, they need microorganisms for their growth. Oxygen is also essential. Alternative air can be bubbled through the reactor. Once again, suppose if it is enter into the oxygen, so it automatically, you know, opposite, the carbon dioxide will be released. The carbon dioxide also will release means it comes up. It will be eliminated in the another way. Alternative air can be bubbled through the reactor. That is also very, very important. And second one is if you look at the three figures closely, you will see the bioreactor has, and normally each and every bioreactor must have 3 figures. Okay, don't forget, this is very, very important, my dear student. One is called as agitator system. 3 figure is very, very important. One is called as agitator system. Second one is called as oxygen delivery system. And third one is called as foam control system. A temperature control system, pH control system, and sampling port so that small volume of culture can be withdrawn periodically. It is also used for the next culture. Okay. So just like a curd, we are taking curd for small amount for the next preparation, isn't it? So likewise, here also there is a 3 figures is very, very important for the bioreactor. One is called as agitator system, oxygen delivery system, and foam control system. Sometimes they may ask the questions, which one of the following is not related to the bioreactor? Likewise, they may ask the question, you have to tell the exact answer. 3 is very, very important: agitator, oxygen, and foam control system. Okay, so this is also important, my dear students. So don't forget. And see that this is the bioreactor. Already we discussed all the part. Okay, so here there is a, what is that? Mixer, isn't it? So this is a very, very sterilization process. After that, sterility also comes out here. Okay, and then another important process is called downstreaming process. Okay, downstream process means after the completion of the biosynthesis stage, completion, the inner side, all the substrate molecules are utilized by the microorganisms. Okay, it will be eliminated. We cannot be able to remove from upper side. Okay, only it maybe comes out from the lower side only. That's called the downstream. See that after completion of the biosynthesis stage, after the production of the lactic acid, after the production of the ethanol or butyric acid or penicillium or any other molecules, whatever it may be, methanol, ethanol, whatever it may be, okay, isolated from the alcohol also, ethyl alcohol, propanol, whatever it may be, all the components, it may be isolated through the downstream process. After the completion of biosynthesis stage, the product has to be subjected through a series of processes before it is ready for the marketing as a finished product. Okay, you know a little about distillation process. Some of them are used for the distillation process. Then it will be pure form. Okay, commercial. Take an example of any one of the Dasmark product. Okay, whatever it may be, alcohol product. It is also a similar things. The process including separation and purification, which are collectively referred to as the downstream process. That is very, very important. Okay, separation and purification. Okay, this is also very, very downstream process. Okay, next one. After the product is formed, either in the free secreted as associated with the cell, the next process is to collect the product from the cell. Okay, collect the product from the cell. No specialized process is required to collect the secreted product, but several methods have been developed to release the product through cell disruptions. Once the product is collected, additional extraction and purification steps are needed to develop final product for applications. Okay, it will not directly, they will not use it after the commercial preparations. They also undergo the further purification extraction process before applications. So that is very, very important, my dear students. Okay. And then see, the product has to be formulated with the suitable preservative. After the preparation, after the purification, separation done, have to add some preservative. Take an examples of any one of the jams, bottle. Okay, bottle prepared food products, whatever it may be, you have to add some preservative. For example, Pepsi, Coca-Cola, whatever it may be, and fruit product, even take an examples of milk. Milk is also there used to H2O2, hydrogen peroxide. It is not good for the infants. At the same time, and fruits already Kisan and whatever it may be, that fruits also, a jam. Okay, there is a preservative, sodium benzoate. And food products, take an example, any cool drinks, Okay, they were add some pesticides, sometimes herbicides like that. They were used. That's why they were banned so many times. Okay, even take an example, some noodles. Okay, noodles also they were used some lead. Lead is not good for health. Okay, so such things. With the suitable result, such as formulation has to undergo through the clinical trials as a case of drugs. Suppose if it is drugs, it undergoes the clinical trials. That is very, very important. They were used for the, they will not use the product direct to the human beings. They were used from some trials. For example, suppose if you are introducing any one of the antibiotics, even take an example, COVID vaccination. After the invention of vaccination, they introduce only mammals, animals, okay, not for direct to the human being. So after that stage, only they introduce to the market as well as the human beings. They will not directly introduce to the human beings. Okay, strict quality control testing for each product is also required. Not only that, after that, strict quality control. Quality should be pure forms. 100% quality would be introduced. Suppose if it is less than that, already you know about it. Some of the childs also drinks, cool drinks. Okay, they were dead. That thing's also given, isn't it? My dear students, the strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. You know what about that? Some of the fruits, some of the liquids, okay, milk as well as a food product, you may be each and every one, they are having a special trimming process. Okay, so that is also very, very important. And the next we'll pass on to the next product recovery of downstream process. The choice of recovery process based on their following criteria. So then only you will get the product very clearly. First of all, the intracellular and extracellular location of the product is very, very important. Intracellular and the extracellular location. The concentration of the product in the fermentation broth, concentration is also very, very important. Okay. And physical and chemical properties of the desired product. Suppose we are taking the milk, you know very well about the milk product, physical property, chemical property. Physical means you know what about. It's a liquid nature, white color. Okay. So how many percentage of white color? Okay, that's all. And chemical means what are the content percent in it, whether the fat percentage, how much is the protein percentage, how much is there, calcium, and other water molecules, how much is there. Likewise, you have to analyze each and every one. Okay, that is also important things. And then the intended use of the product, So this is another one. The minimal acceptable standard of the purity, that is very, very important. Already, you know, any one of the industries, there is a standard is there. For example, any one of the product, take an example, Horlicks, Boost, whatever in it. Okay, there is a standard procedure is there. This much containing this much of protein, this gram containing this much of vitamins, minerals, like that. Okay, that is also very, very important. Standard is also there. This much of standard is also maintained by the food companies. The magnitude of the biohazards of the product or bath, magnitude, biohazards of the product or broth. Suppose when you are using the microorganism as a broth or product, it will not produce more amount of biohazards. Hazard means dangers, harmful effect it will not produce. That type of broth you have to use it. The impurities is the fermented broth. That is also another one. The marketable price for the product. These are the important things. Very, very essential, my dear. Next one is the whole range of biochemical separation and purification technique is available, such as drying chromatography, solvent extraction, and distillation. These things already I told you. The whole range of biochemical separation and purification technique is available, such as the biochemical technique is essential. That is all I already told you. Chromatography technique. Listen, it chromatic, paper chromatic, different types of it. Indeed, there is a subdivision is also there. Okay, and also these are very, very important. So that's all about my dear these classes. Okay, next part is what is the evolution part. We will see the remaining things on the next classes. Here you must note it down. 5 questions have been reported here. One is what is a match the following, column 1 and column 2. Bacillus thuringiensis, Thermosaccharicus, Aquabacterium, Timifaciens, and also Salmonella typhimurium. Okay, already these things also already very, very important. Bacillus thuringiensis, you know about that DNA polymerases. Taqua cryogenes, Aquapaticum timifaciens. Okay, so this is nothing but the cloning vectors. Salmonella typhimurium, construction of the first rDNA molecules. This is the one. Next one is, okay, see this one. The specific palindromic sequence which is recognized by the E. coli. Okay, which one is the correct one? This one, you know, the first one is the correct one. Okay, GAA-TTC. Okay, see GAA-TTC. So this is very, very important one. Others it will not come. These are all a direct question, my dears. Don't forget. And the next one is the in mixture DNA fragments are separated by, okay, DNA polymerase chain. DNA fragments are separated. Fragments means already we studied polymerase chain reaction only for the RNA DNA amplification. We will produce more amount. It will not separation. Bioprocess engineering, it will not come. Restriction digestion, it will not come. Electrophoresis, that is very, very important medicine. Electrophoresis is the answer. It's the correct one. Okay. Electron, we are using the electron to separate the molecules in very minute quantity also, particularly DNA, RNA, protein, or whatever it may be, that things. So the electrophoresis is the correct answers. Okay. What are the next one? In recombinant DNA technology, antibiotics are used, okay, as selectable marker to keep medium bacteria free and to detect the alien's DNA to important disease resistant to the host plant. See that in recombinant DNA technology, antibiotics are used. Which one you know? So, to keep the medium bacteria free, to detect the alien DNA, to import a selectable marker. Okay, which one is the correct one? Will you please tell me? The important disease resistant to the host plant. That is very, very important. Next one is the first discovered restriction endonuclease that always cut DNA molecules. Already you know, but which one is this one? Adenose deaminase, it will not come. Thermostable DNA polymerase, it will not come. Isn't it? Hind2Na is the correct one. Already we studied it. This is the first DNA molecule discovered, isn't it? So Hind2 is the correct one. Okay. What is the next one? During the purification process for recombinant DNA technique, addition of the chilled ethanol precipitate out. Which one is precipitate out? Polymerase enzyme, it will not precipitate out. Histone is a protein molecule, it will not precipitate out. RNA is not a precipitating out, but the DNA molecules only. DNA technique, DNA only precipitate. So this is the correct answer. Okay, my dear student. So see here, sixth one. Okay, so these are all not answer. Okay. So that's all about my dear students. Today class is over, but particularly the lessons also finished. Okay, the biotechnology finished. We will see the next lesson, biotechnology, application of the biotechnology. We'll see the next class. Thank you so much for the participation. Thank you. Take care. Bye. Good night. Happy Pongal.